Question number 1.
The primary purpose of chromosome banding techniques. Select the correct option. A. To determine chromosome length. B. To study mitotic division. C. To measure DNA content. D. To create a visual pattern for identification and diagnosis.
Question number 2.
What staining technique is commonly used for visualizing chromosomes in karyotyping? Select the correct option. A. Ethidium bromide. B. Kumasi brilliant blue. C. Crystal violet. D. Giemsa Stain.
Question number 3.
Which of the following is a crucial factor in primer design for PCR? Select the correct option. A. Specificity for the target sequence. B. High GC content. C. Low melting temperature. D. Short length.
Question number 4.
cDNA is produced from. Select the correct option. A. Protein. B. mRNA. C. DNA. D. tRNA.
Question number 5.
The enzyme used in formation of cDNA from mRNA is. Select the correct option. A. Helicase. B. Transcriptase. C. Gyrase. D. Polymerase. Note: correct term is reverse transcriptase. Options may transcriptase, DI.
Question number 6.
In real-time PCR, what is the purpose of using a fluorescence-labeled primer? Select the correct option. A. To increase primer specificity. B. To monitor DNA amplification in real time. C. To decrease the melting temperature. D. To enhance primer binding.
Question number 7.
The primary objectives of isolation workflows are to stabilize RNA molecules, suppress, and optimize yield through appropriate storage and extraction techniques. Select the correct option. A. Proteinase. B. Transcriptase. C. DNases. D. RNases.
Question number 8.
Which enzyme is commonly used to synthesize cDNA from RNA templates? Select the correct option. A. RNA polymerase. B. Reverse transcriptase. C. DNA polymerase. D.
Ligase.
Question number 9.
The primary objectives of isolation workflows are to stabilize RNA molecules, suppress, and optimize yield through appropriate storage and extraction techniques. Select the correct option. A. Transcriptase. B. RNases. C. DNases. D. Proteinase. Select the correct option.